Good afternoon once again. I'm welcoming you to another topic and another chapter of codominance, which we are to explain through some diagrams as being shown on the screen or on the board there. Codominance, it is another form of expression that we always find again as a typical example or as a typical area of concern in metric exams. Firstly, I would want to define what is codominance. It is a form of expression where two alleles of a gene in a heterozygote produce different effects. Two alleles of a gene in a heterozygote produce different effects, and the effects are seen in the heterozygote. So what is happening here is that the two alleles of a pair are both expressed in the phenotype. The two alleles of a pair are both expressed in the phenotype of that heterozygote, and the individual has got characteristics that are coded for, which are coded for by both alleles. We have got a typical example of allele A and allele B, which code for blood type AB as a typical example of codominance. We are saying allele A and allele B, which are a typical example which represents blood type AB. They are, or it shows a codominance. So I've given this, I come up with this diagram that is going to show inheritance of different blood groups in a family. The diagram that is shown there, it is actually going to explain, it's actually used to explain the inheritance of characteristics in a family, which we would always see that the horizontal lines are representing individuals who are maybe making a family, who we say that they are making a family, and then the downward line, the vertical lines, are showing descendants, which are children from a certain family. So when the horizontal line is joining the two people, they are a couple, or they are the parents, which are joined by a horizontal line. They are indicating to say that those people are they are a family, and then which are also going to show the descendants that are shown by the vertical lines that are going down, that represents the children of those families or of those parents. So let us quickly first of all answer these questions. After answering these questions, we would further explain and further identify other areas of concern with regard to blood groups as an example of a codominance, which we say that blood type AB is an example of a codominant that is shown by a combination of allele A and allele B. These alleles are always dominant alleles. Hence, when they combine together, they represent, or they are going to be expressed, they are going to be shown in the phenotypic expression of the individual who has got these both alleles coded together there. So let's answer the questions that are here. As we are answering them, we are going to be also further explaining reasons why do we say that that is the answer for each given scenario that is there. So the first question is saying, the first question in our on our board here is saying, name the type of diagram that is shown there. What is this diagram? Question 1.4.1. Name the type of diagram that is shown. The type of diagram shown. What diagram? What is this question? 1.4.1. What is this diagram? It is called a pedigree diagram. We are saying this is a pedigree diagram. Pedigree diagram. It's a pedigree diagram, this one, which is shown here, pedigree diagram. It's a diagram that shows chances of inheritance. It shows the inheritance of characteristics in a family what it does is that it is showing the possibilities of inheritance of situation of conditions that can be inherited in a family. So what is happening is that like I said at first, that there are horizontal lines, vertical lines that are descending from a certain line or a certain point of contact where two are joined together. For example, in this question, we can say RT and Chris. RT and Chris can be given as parents. And then we've got Anne and Lina that who are also on our diagram here. RT and Chris can be parents, and then we are saying this horizontal line is showing that Anne and Lina are children who are born from these parents. So based on their genes of the parents, they are going to give children who can have certain conditions or who can have certain characteristics, and their characteristics are those that they have inherited from the parents. So then what we are saying is that it's a pedigree diagram that is shown there. That is question 1.4.1. Question 1.4.1. It's a pedigree diagram that is being shown there. The next question is saying, give the number of alleles that controls blood groups. The number of alleles that controls blood groups. When we are into blood groups are controlled by 3 alleles. Question 1.4.2, the number of alleles that controls blood groups is 3. Is 3 blood groups that controls, three alleles that controls blood groups, 3 alleles. And then maybe sometimes the question would want you to say, name these alleles that are there. We have got allele A, or we can have allele B, or we can have allele I. So what we are saying is these 3 are the 3 alleles that controls the blood groups. So we are saying allele A, allele B, allele I, they are, or these are the blood alleles, or the 3 alleles that controls the blood groups there. Next question is saying, how many generations are represented in the diagram? How many generations are represented in a diagram? The generations that are represented in the diagram, 1.4.3. How many generations? If we can look in our diagram here, we are saying generations. When we talk about generations, we refer to people who have lived in the same time. We have lived at the same period of time, which would say they are in the same age group. So we refer those as generations. Referring to our diagram here, we can see that they are, we can say this one, we can have also 2, and then we have got 3. All of these people, we are saying the first part, which is 1 there, which has got the names, or which have got individuals, says T and Chris, we are saying the generation. And then we have also Bob, Annie, up to Vusi, they are also a generation. And then we have got also the last group, which has got these individuals whose blood groups are not shown. So how many generations are represented in the diagram? There are 3 generations. There are 3 generations that are represented there, as I've shown there, who are represented or who are shown by the light color orange yellow that I've highlighted with on the diagram there. Those are the 3 generations that are shown in the diagram, or on our question there. Those are the 3 generations that are shown there. And then the next question is asking, or is saying, question 1.4.4. Question 1.4.4. Lina's genotype is A, heterozygous. State all the possible genotypes of Vusi. The possible genotypes of Vusi. Vusi is this one. What are the possible genotypes of Vusi? When you look there, we are saying our VUSI is this one. This is our VUSI here. So VUSI, what is the possible genotype of VUSI? Genotype, we said, is the genetic makeup that can cause us to have them having VUSI and Lina are parents, and they have got children whose blood group is A and the other one is blood group O. So the possible genotype that can arise, or that can be for VUSI, what can it be? Having that, we have got one child who is who is uh who is blood type A and the other child who is blood type O. One, we are saying there is a possibility of saying VUSI. VUSI can be blood type number 1.4, 1.4.4. The blood type of VUSI, we can have VUSI having the blood type 1.4.1.4.4. VUSI's blood type, we can have VUSI having blood type A heterozygous. We can have VUSI having the blood type A heterozygous, because in that case, there is a possibility of an I there which can combine with Lina to have I there. So we can have VUSI having heterozygous A. It can as well be possibly having heterozygous B there. We can say VUSI can as well be able to have heterozygous B, as to say that can as well be another possibility that can be there, to say if VUS is heterozygous B, we can as well be able to have Lina having blood type O there. And then eventually again, we can have VUSI. VUSI can also be blood type. It can as well be blood type O. It can as well be blood type O, because we are saying having Lina with that blood type there, the genes or the inheritance of O in either of the, in one of the children, which is there, is that we can have O from that heterozygous, or with that heterozygous A, heterozygous B, also as O, can be another possible genotype of VUSI there. That is number 1.4.4. Next question is saying, give the genotype of Bob. Give the genotype of Bob. Bob's genotype, we can check it nicely. The genotype of Bob. Bob is a child who is born. Bob is a child who is born from parents who are both blood type O. So if parents are both blood type O, there is no any other way that Bob can have another blood types. So what we are saying is, give the genotype of Bob. The genotype of Bob can only be what? The genotype of Bob can only be 1.4.5. Bob's genotype 1.4.5. We are saying Bob's genotype, the genotype of Bob there, the genotype of Bob at 1.4.5. 1.4.5. Bob can only be the blood type, or Bob can be blood type O, or which is I. Blood type O is the genotype, which is the genotype is I there. The genotype of Bob can only be that. Can only be that one, which is O. And then give the name of the individual which displays codominance. We said codominance is when a person or the individual has got a combination of A and B allele. So 1.4.6, 1.4.6, 1.4, 0.6. Which individual can be blood type AB? This individual, if you can see from our pedigree diagram here, the individual who can be blood type AB, which individual can be blood type AB? Who is that person? If you can look at our pedigree diagram there, we are saying the person who can be blood type AB can be N. N can be, is the person who is blood type AB, where we are saying N and Bob can have a child who is blood type B, who is blood type also A. So question number 1.4.6, the individual who is blood type, who displays codominance. Number 4.1.6, the individual who displays codominance. We are saying this individual can be N. He is the person, or she is the person who can be blood type AB based on our pedigree diagram there. So pedigree diagram, it actually shows these possibilities of inheritance of characteristics. So it comes in this way, this question of blood typing. That is how sometimes it comes like, but it is not the only way that it can be, but we just need to further explore this question or this answer or this situation. You know, as we are looking at blood types, there are other questions that can as well arise there, which can say, how many blood types do people have when we are using a blood AB or blood typing? Blood typing, we are saying we can say how many blood types or how many blood groups, number of blood groups, uh, number of blood groups that we have using we are saying based on ABO blood typing, based on ABO blood typing. How many blood groups do we have? We have got blood type, we have got how many blood groups? We are saying these blood groups, there are four, there are four of them. We have got four, there are four blood types. Let's name them. We can have blood type, we can have blood type A, we can have blood type B, we can have blood type AB, we can have blood type O. And then these are the four blood types that we can have there. There are four blood types that can be there, four blood types that can be produced or when that we can have, talking about ABO blood typing. And then in this case, we need to understand again, or I would want to give you another table that is supposed to be understood there when we are under blood typing. When we are in blood typing, we sometimes also need to understand the genotypes, the alleles present, the antigens that can be produced in the blood, the individuals who can give blood to others and who can receive blood to others. So I would want to present to you this table that can be used also to further explain and further make this topic of blood typing complete and thorough. Blood typing, we can have a table that we can give here. This table, I'm going to just quickly draw it here, which is going to be having our blood types. I'm going to quickly draw this table, and then we can be able to use this table for our explanations and discussions. I want you to see this table because it is quite important for us to know it, so that whenever we meet it again, we won't be having problems. So we have got blood group. So in the first column, we have got blood group. blood group, and then we are saying is the phenotype of the blood group, and then we are saying the alleles that controls this blood group. Obviously, when we are there, we are referring to genotype. Genotype, the alleles, we are saying it is the genotype, and then we also have what is called antigens that are produced by each blood type. The antigen produced, and then we can say the individual can give blood to certain people. Can give blood to, and then eventually can receive blood from. Receive blood from who can give and who can donate blood to who. So with our table here, I'm just going to quickly summarize this one. We are saying we have got blood type, like I said, we have got 4 blood types there. So let us start with blood type A. Let's start with blood type A. So the phenotype, obviously, we are saying it's the blood type, it is blood type A. And then the alleles that controls blood type A can appear in different ways. We can have blood type A as like saying it can be our A can be like this, which is A dominant, or we can have our A as like that, to say a person who is having that is having, it is blood type A, which can be either an A homozygous or A heterozygous. And then we are saying what antigen is produced. This individual produces antigen A, and then maybe other people would want to understand to say, what is it that you are calling antigen? It is a substance that an antigen, we are saying it is a substance when introduced into the body stimulates the production of antibodies. A substance when introduced into the body stimulates the production of antibodies. That is an antigen. We have got, we are saying production of antibodies, which, for example, the proteins on the surface of the red blood cells are a good example of antigens. So what happens is that antigens affect the way in which blood donation would take place. What is happening is that when a person receives blood or when a person is given blood, the blood must be able to mix without causing clumps. If it causes clumps, it causes the person to have problems that will arise where the blood cannot circulate, and then when it not circulate, therefore it will cause the death and closure of the blood vessels. Hence, we are saying the patient, the individual or that person who has received the blood can have problems that might cause the person to die or to be paralyzed. So what we are saying is that we have got antigen A that is produced by blood A, and then this person, we are saying this person can give blood to people with blood A or people with blood AB because he produces antigen A. So can give blood to people this person can give blood to people who are blood A, or can also give to people who are blood AB. The idea is that the people with blood groups already, these blood groups, what we are saying is that they already produce the antigen A, so their blood can recognize the new antigen A that is produced by the blood that is being donated to this person. And then he can as well can receive blood from where? he can receive blood from people who are also either blood A or who is blood what? Or who is blood A or O. This person can receive blood from people who is blood A or blood O. So what we are saying is that blood A genotypes are those ones that I've given you there, and then it produces antigen A. And then antigen A, we are saying it is a substance which when it is introduced in the body, it causes the production of antibodies in the body, which we say the protein substance or the protein molecules that are found on the red blood cells. We are saying those are examples of antigens. Let's have also the next example, which is uh, blood type B. The next example is blood type B, which we are saying B can also be, we can have B like this, or can be this B, which is equally the same again to say a person can be blood type B homozygous, or can be blood B, which is heterozygous B, and then also can produce antigen B, there. And then what is this person? This person can as well give people to individuals who are blood B or who is blood AB. And then similarly again, can receive blood from people who are blood O, blood B, or who is blood O. And understand that blood O does not produce any antigen. There is none of antigens that are produced there. That is why you see individuals who are blood A and blood B can receive blood from somebody who is blood type O. So because there is no antigens that are produced by O. And then our next blood type that we need to look at, the next blood type that we need to look at is blood type AB. Blood type AB, what are the alleles that it produces? What are the alleles that are there that produces blood type AB? We are saying this one, as we have said, it is a codominant blood type because A and B are alleles that are equally dominant. And then when we talk about the blood alleles or the genotype, it is always supposed to be allele A and allele B, which is like that. And then the antigens that are, what antigen produced there? In this case, obviously, it is AB, meaning to say we would have also antigen. We have got antigen A produced and also antigen B produced at the same time. Antigen A and antigen B is produced there. And then can give blood to who? This person can give blood also limited to people who are blood type AB, because antigen A and B are produced at the same time whenever the blood AB is there. So they produce antigen A and B, which are also produced there. Hence, we are saying they can cause blood clumping or blood clotting, which would cause the clumps that will form the clumps that will form, the blood then forms clumps of dead red blood cells. This might also kill the patient. So this person can also receive blood from people. He can receive blood from people who is blood A. He can receive from blood A. He can receive from blood B. He can receive from blood what? B, because we are having antigen B produced. It is already recognized because B is already there. And then we can also receive blood from AB and O. And O finally. We are saying can receive blood from either A, he can receive blood from O, can receive blood from AB. Those are the different blood types which this individual can receive blood from. And then the last blood type that we said we have is blood type O, which you can see from the list of blood types that we have given there. We are saying we have got the last blood type, which is blood type what? Which is blood type O. Blood type O, the alleles that it produces, or that the alleles that produces blood type O is I recessive. There is that the allele that produces blood type O, and then that is its genotype. And then does it produce any antigen? There is none antigens that are produced. There is none antigens produced by O here. You are saying O produces none. No antigens are produced there. And then can give blood to who? Who can this person give blood to? Can give blood to all the other people or to all individuals. You can give blood to people with blood A. You can give to people with blood B. You can give blood to people with AB, and also can give blood to and O. And then this person can receive only blood from people who are blood O. Can only receive blood from people who are blood O. As you can see, the condition is that the individual must only receive blood which can be able to mix without causing any clumps, without causing the blood to clump in the body. Because whenever they are there, remember we said that if the patient receives, or if this person is a patient, therefore we say he receives the blood which has got certain antigens. We are saying his white blood cells do not recognize the antigens because the antigens are not present in his red blood cells. Hence, he has to receive blood only to people with blood O, because his blood type does not have any antigen in it. That is why he should not receive blood from any of the other three blood types there. That is the thing. That is the idea. That is the point in this context. So we are saying these are the blood types and their antigens and the individuals who can receive and give blood to. I'm sure that you also understand that blood. I'm sure that you can as well understand that what is this thing to say a person who donates blood is the blood donor. That person who donates blood is called a blood donor. When blood is donated, it is always classified and then it is put into a blood bank where it is kept for any other uses. I'm sure that we have heard of South Africa Blood Services and stuff. So therefore we are saying when blood is donated, it is classified and then it is kept safe for any other uses whenever patients would need a blood transfusion. So that is what is happening there with regard to blood groups and the blood typing. And I'm sure we can as well have what is it? We can as well have our next question, which is going to be another part of this question, which is going to be just showing us, which is just another part of this question, where we are going to talk about the last part of this question of our question paper that we are discussing now. Let us just quickly discuss it so that we can be able to finish it. We can be able to finish it first, and then the last question that is here is an investigation that is done. An investigation that is done. That is the last question. Learners conducted an investigation. Learners conducted an investigation, which is given there. Learners conducted an investigation to determine which blood groups was most common in their community. They collected information about the blood groups of 200 blood donors in each of the 3 blood donor clinics in their community. They did not include first-time donors in the investigation. The pie chart below shows the result of the investigation, which is there. The pie chart shows the result of the investigation, which is given there. So we are saying the first question, without wasting time there, we are saying we have got the representation, 25% blood A, 15% blood B, 5% blood AB, and blood O has got 55%. Has got 55%. And then the question is saying an investigation, conducting to determine which blood group was the most common in their community. So our first question is saying, state the aim of the investigation. The aim of the investigation, I'm sure that you can see from this question 3.1, the aim of the investigation 3.1, state the aim of the investigation, state the aim of the investigation. The aim is to determine which blood group is most common in the community. It is to determine the most common type of blood group in the community. That is the aim, as it is shown there, to determine which blood group was most common in the community of these learners. So 3.1, question 3.1, 3.1, question 3.1, what is the aim of this investigation? We are saying to determine the most common blood group in the community. The learners wanted to determine the most common blood group in the community. This is actually shown also there, as the pie chart indicates the percentage ratios of each group. The percentage ratios of each group is representing the most common type, the most common blood group there, which is to say the one which has got the highest representation or the highest percentage is the one that would say is the most common one, is the one that would say is the most common blood group there. Is the one that we can say is the most common blood group. Next question is saying, that was 3.A. This is A, and question B, okay, 3.1.2, 3.1.2, question 3.1.2, 3.1.2 is saying, stage 3 planning steps to consider when conducting this investigation. The learners were to conduct an investigation on the blood groups. So what are the planning steps that they were supposed to consider? The planning steps that they were supposed to consider there. There are quite a lot of them, but we are just going to list but just a few of them. We're just going to list just a few of them. Stage 3 planning steps that are supposed to be considered. 1, obtain permission from the school or clinic to conduct the investigation. They were supposed to obtain permission. 1 is getting permission, obtain permission, which is from either the school or a clinic. They must get the permission from the school or the clinic. That is first planning step when they are to do this investigation. Decide on the sample size. Deciding on the sample size, obviously, as you can see, groups of 200 donors, we are saying blood donors, so deciding on sample size, which we are saying the sample size must always be big enough, so therefore the reliability of the results can be found. can be correct. We don't need to use small sample sizes, because the smaller the sample size, the higher the chances of the results to be not correct. And again, we can also talk about decide the method of data recording, or decide on the method of recording the results. When an investigation is to be done, we also need to decide on the data, on the method of number 3. We are saying method of deciding on the method of recording the results. Which method was going to be used to record the results? It must be decided before the investigation is done. Before the investigation is supposed to be done, also it needs to be decided there. And then another point, number 4, we are saying the planning steps. Number 4, which we are just giving planning steps to consider when conducting an investigation. We are saying deciding on the time or date to collect the data from the school or clinic. It's a planning step to say when were they going to do the investigation, deciding on time or date to collect. We are saying to collect the results, collect the data. Quality the data from school or clinic. From school or clinic. From school or clinic. From the school or clinic, that must be decided as your school or clinic. The date or time must be decided. Last but not least, we can also talk about decisions were supposed to be made on the environmental conditions again, which we can as well give to say the learners, they might need also to take decisions on environmental conditions. They should also decide on the environmental conditions that they are going to use or that are going to be affecting their investigation. Number 5, deciding on environmental, deciding on environmental conditions. I'm sure this can be enough. And then question next is saying, state one way in which the learners ensure the reliability of results. Number 3.12, 3.1, state one way, 3.12b, state one way in which the learners ensure the reliability of the results. What we are saying is, how do you see the learners ensuring the reliability of the results? They collected information about the blood groups of 200 blood donors in each of the three blood donor clinics in their community. So what we are saying is that how do they ensure that the results were correct? One, we are saying included 3 samples. They included we are saying included 3 samples That is to mean to say that they were trying to improve the reliability of the results, including 3 samples that are there. The first point, they included 3 samples. It was to ensure that the results are correct, included 3 samples, which can be 3 samples of more or more. We are also saying they also used 200 donors per clinic, 200 donors per clinic, which is something like 600 donors in total. So having a large sample size is another point also that can be given there. Large sample size of 200 donors per clinic. $100 per clinic is also a good way of ensuring that the results are large sample size of 200 donors per clinic. It's also a good idea, or it's a good measure that was taken to improve the reliability of results. And number C, give one reason why they did not include the first-time donors in this investigation. Why should we not include the first-time donors in the investigation? Number 1.3.1, 1.2b, C, question 3.13c. Why did they not use first-time donors? Question C. First-time donors, we are saying they were not included because we are saying their blood type, their blood groups are not yet known. That can be the reason to say we cannot use them, or they were not used. Their blood groups were not yet known. You can also say that their blood group was not captured in the database. Not in the database also. It can be the reason why we cannot include them, or why they were not included. Their blood group was not also in the database. Calculate the number of participants that had blood group B. Show all your working. People who had got blood B. In this case, we are supposed to see blood B, calculate the number of participants that had blood B. Blood B percentage ratio or percentage representation is 15, and then we are saying 15% of the people that got blood B. So what we are saying is that it is 15%. It is 15% times, 15% of the total participants that were there. It is 15% of the total participants who were there, which is question 3.13. 3.13. Question 3.1.3. We are saying it is 15%, 15 over 100 times the total number of participants. The total number of participants, there were how many? It was 600, because 200 per each, or 200 from the 3 representations, which is going to be saying times 600 over 1. The 600 is 200 times 3. That is where the 200 is coming from. I mean, the 600 is coming from. So here we say that this is equals to 15 over 100 times 600 over 1. So how many are those people? We put it in the on. I mean, we just press it on our calculator there. That would give us something like what is this? 90. That would give us 90 participants. That will give us 90 participants. Well, which is 90 participants. Blood B is equals to 90 participants. Blood B is equals to 90 participants. 90 were from blood B. And then question 3.14, name the blood group which has only recessive alleles in the genotype. Name the blood group which has got only recessive alleles in the genotype. I'm sure that from the first question that we have answered, or from the question on top there that we have answered, we are saying the blood group that has got only the recessive genotype. I'm sure this can as well be seen, or it can be seen from our first question that we've been calculating there. The blood type which has got only the recessive, the blood type, the blood group which has got only recessive alleles in the genotype is blood type what? 3.14. 3.14. 3.1.4. It is blood type. It is blood type O. Blood type O, without even seeing, blood type O is the only blood type which has got only recessive alleles in its genotype, because it can only form O when I and I have been joined or have close together. That is 4. And then B is a result of codominance. Codominance, we have already given it again in our first part of the question there, which we said that codominance, it is whereby allele A and B join together. So codominance is always given by this is A, and then B, it is blood type. It is blood type, blood group AB. Blood group AB is codominance. And then lastly, give all the possible genotypes of the blood group represented by 25% of the donors. 25% of the donors is there, which is blood group A. Blood group A is 25%, and then the question is saying, give all the possible genotypes of blood group represented by 25% of the donors. Question is question 3.5. 3.5, question 3.5. Question 3.5, which is blood type A. We're saying blood type A 3.15. 1.5. It can be blood type A, which is A homozygous, or it can be A heterozygous. And then, having said that, we have answered all the questions that this blood group question is asking. We have answered all the questions there. We have answered all the questions. I'm sure that in terms of blood grouping, we have gone through all the possible angles which can be asked there. So thank you very much. I hope that you are going to understand and get a good preparation for your exams. all the best, and keep reading and keep watching for the next coming uh, presentation, which is going to be anytime soon. Thank you very much, and enjoy your studies. Thank you.